Welcome to the debate. Today we are diving into, well, something quite ancient, yet really relevant right now, sourdough fermentation. It's kind of exploded, hasn't it? Not just, as something tasty to eat, but as a topic for serious science and, interestingly, for hands-on learning. Our sources really highlight this kind of dual nature, the measurable health benefits on one side, and its use as a living model for biology education on the other. And that sets up our question for today. Where does the main significance of sourdough truly lie? Is it in those proven, quantifiable health boosts, or is it more about its unique value as this tangible, living classroom for teaching pretty complex science? So I'll be arguing that the greatest impact really is in those physiological and health benefits that we can measure.
And I'm coming at it from, a slightly different angle. I certainly don't dismiss the nutritional side, that's clear. But I'm going to focus on the, let's say, enduring, educational, ecological, and maybe even philosophical value you get from the process itself. For me, that sourdough starter is just an incredible, accessible, living lab, a really powerful way to build scientific understanding and, frankly, patience.
Okay, so my core position, it really centers on sourdough being a practical nutritional tool. The science backs this up. It's shown to genuinely enhance health and actually help combat some of these modern metabolic diseases, mainly through how it transforms nutrients, this biotransformation. We've got some pretty compelling data that takes sourdough way beyond just, keeping bread fresh longer. First off, fermentation significantly lowers the glycemic index, the GI, of bread. What that means is it stops those really fast, damaging blood sugar spikes you get from, say, typical white bread. You get a much slower, more sustained energy release instead. And critically, the process cuts down on FODMAPs, that's a mouthful, fermentable oligosaccharides, disaccharides, monosaccharides, and polyols. These are essentially carbohydrates that many people struggle to digest, causing significant gut issues. The acidity from the bacteria just breaks them down really effectively. What's more, that long fermentation time, it actually makes essential micronutrients like minerals, vitamins, those phytochemicals more available to our bodies. It does this by neutralizing things called anti-nutrients like phytic acid. Phytic acid naturally occurs in grains and it binds to minerals like iron and zinc, stopping us from absorbing them. The bacteria in sourdough produce enzymes that break down this phytic acid, basically unlocking those nutrients.
Mm-hmm. And maybe the most convincing part, for me anyway, is the specific research done on diabetic models. Certain sourdough breads were fed to diabetic rats, and the results were quite striking. Significant improvements in cognitive issues, lower blood sugar, and less oxidative stress and inflammation. This really supports the idea it could be useful against conditions some researchers are now even calling type 3 diabetes, thinking about Alzheimer's linked to insulin resistance in the brain. So the benefit isn't just theoretical, it's confirmed right down at the molecular level. That's definitely a strong case for the nutritional side of things, the outcomes. But I'd argue that those outcomes, important as they are, they're kind of the end point of a process whose real lasting value is maybe more intellectual. The enduring importance of sourdough, I think, isn't just in the final loaf, but in how it functions as this amazing living laboratory that's accessible to pretty much anyone. You just need flour and water. It's this fantastic tool for teaching core ideas in biology, ecology, even data science, but in a really practical, hands-on way. I mean, the starter itself is like a physical microbial zoo, right? It lets students, or really anyone, these citizen scientists, track really complex changes in a biological community in real time over, say, a couple of weeks. We ask them to track things they can actually measure, like how high the starter rises. That gives a direct visual link to the tiny yeast inside producing CO2 gas. And they also measure the pH, which directly shows the activity of the bacteria, making lactic and acetic acids. This whole process, it doesn't just drill facts, it vividly shows ecological succession. They literally watch as the first microbes, the pioneers, are gradually replaced by the stable, specialized climax community that can keep going indefinitely. Even just seeing that layer of liquid, the hooch that forms on top when a starter gets hungry, that's a tangible lesson right there. It signals the yeast are stressed to making alcohol, as a different way to get energy. It's basically science in a jar, easy to grasp.
Okay, I see why you emphasize the accessibility, the learning aspect, But the sheer efficacy against chronic disease, that provides a quantifiable usefulness, a clear, measurable impact on, well, on people's lives and health. If we only focus on the educational metaphor, I think we risk minimizing the real-world, potentially life-changing results that this advanced research has uncovered. I mean, the study showed feeding diabetic rats that optimized sourdough bread actually significantly turned up the expression of beneficial genes. We're talking specifically about BDNF, that's brain-derived neurotrophic factor, and NRF2 in the hippocampus, part of the brain. BDNF helps nerve cells grow and survive, basically boosting brain health, while NRF2 is like the body's master switch for antioxidant defenses. And at the same time, it turned down negative genes like GLUT2 and PEPCK in the liver, which are involved in making and releasing glucose.
This molecular proof of, protecting the brain and improving blood sugar control isn't just symbolic. It's hard evidence of really deep biochemical changes that offers a potentially life-altering benefit that I still argue fundamentally outweighs the, let's say, conceptual value of a home science project, however interesting that project is.
I appreciate the rigor of that study, absolutely. And those molecular pathways you're describing, they're definitely valuable. They help explain why the sourdough is nutritionally better. But my counter would be that those kinds of results, they're often stuck behind, complex lab techniques, really only accessible in research institutions. The educational model, though, it kind of democratizes the whole inquiry process. It gives accessible, hands-on, long-term learning to everyone, and that in turn actually fuels the larger scientific effort. Think about it. When students or home bakers are collecting data every day on their starter's height and pH for 2 weeks, they are doing genuine citizen science. They're contributing data that helps map microbial communities. It helps researchers understand how things like local environment, temperature, and especially different types of flour, like using ancient grains, emmer or rye or millet, how those affect the way the microbes grow and interact. We still don't know a lot about the complex dynamics in traditional, spontaneous sourdoughs from around the world. And this accessible educational model is actively helping fill in those knowledge gaps. So it's almost ironic, your findings need advanced gene sequencing, but the basic data needed to understand sourdough diversity globally, that can be gathered with simple tools right at home. And that process fosters real scientific curiosity and skills that stick with people for life. And shifting back to that learning environment for a second, the educational value is so deeply connected to understanding microbial ecology. That's a really complex field, right? But the starter makes it instantly relatable right there in the jar. The source material even explicitly talks about concepts like mutualism, positive relationships, noting how certain lactobacillus species thrive together, helping each other out. And conversely, it details really strong competition, where different yeasts are basically fighting over the limited food. Just learning that Saccharomyces cerevisiae, that's common baker's yeast, is a bit unusual, because it actually has a positive, mutualistic relationship with certain lactobacillus bacteria, letting both of them flourish. Well, that provides a concrete, living lesson in how complex ecosystems work, the dynamics of cooperation and competition. Trying to replicate that kind of lesson, that delicate balance and specific interaction, just theoretically in a classroom, it's incredibly difficult. The starter is constantly teaching this lesson of symbiosis and struggle just by existing.
I'm not entirely convinced by that line of reasoning, because if the ultimate goal is functional efficacy, if we're trying to solve those real health problems we talked about earlier, then getting the best practical results absolutely depends on very informed, very specific choices, selecting the right microbes and the right ingredients for nutritional benefit. Just observing for observation's sake, well, it isn't enough to achieve that. Remember, the study showed the best health outcomes came from that specific sourdough bread too. It wasn't just any random mix, it was deliberately formulated with things like lupin seeds, oats, sunflower seeds. And this highly specific blend resulted in dramatically higher levels of fiber, those beneficial phenolic compounds, and unsaturated fats, particularly linolenic acid, which made up, almost 40% of the fatty acids in that bread. This optimization really underscores that the critical factor is smart nutritional design. Yes, it's guided by microbiology, absolutely. But the aim is to create a functional health product. So the microbiology becomes a sophisticated tool for a really important health outcome. Not the main goal in itself. The benefits aren't just generic. They have to be specifically engineered for maximum impact. And look, the need for that long, slow fermentation time that's scientifically validated
precisely because it leads to better products. We know the benefits are directly linked to maturity, to reaching that point where the microbial metabolism is stable and has optimized the nutrient profile. For example, the low pH you get during that extended time encourages the formation of resistant starch. That's a key reason we see the lower glycemic response. This lengthy process is what ensures the maximum breakdown of those anti-nutrients we discussed and the release of bioactive compounds like leucine and GABA. Even the pedagogical materials confirm that the sweet spot for getting the most benefit happens when the starter reaches that stable climax community you mentioned earlier. So the slowness isn't just a philosophy, it's a necessary functional requirement to get the complex biochemistry right, reducing starch digestibility, maximizing nutrient availability. It's a requirement for efficacy. We have to wait because the chemistry demands it, not simply because we're aiming for patience as a virtue.
That's a fair point, and I totally agree the chemistry needs time. But I think you might be overlooking the really profound meta lesson that's embedded in that required waiting, especially in our modern world. Have you considered the philosophical side of committing to that time? Dr. Johnson actually calls this a pedagogy of fermentation, the explicit lessons you learn from waiting, from patience, from finding a rhythm, and from accepting a certain amount of uncertainty. It actively pushes back against what he terms the cultural violence of acceleration. The starter, just by its biological nature, it resists being rushed by technology. You can't really speed up the microbes effectively without messing up the final product. So it forces this kind of mandatory surrender to biological time. And beyond that, the focus it demands, the attention, the presence required for that daily feeding ritual. And learning from the inevitable, failed loaves or starters that just do weird things, that teaches adaptability and resilience way better than any abstract lesson could. These lessons, they go way beyond the kitchen. They apply directly to creative work, to problem solving, to critical thinking. It teaches the deep value of letting ideas prove or mature, rather than always forcing a sprint towards instant results.
Look, while the philosophical angles and the educational metaphors from Sourdough are undeniably very rich and compelling, and I absolutely support its role in things like citizen science, the hard, concrete, molecular evidence just can't be ignored. The proven reduction in anti-nutrients, the boost in nutrient availability, and especially the demonstrated effects against type 2 diabetes and related cognitive decline, that, for me, firmly establishes its primary value as a powerful, measurable nutritional tool on the modern health landscape. Optimizing that microbial community leads to tangible medical benefits, which are becoming increasingly vital in a world really struggling with chronic metabolic diseases.
I agree. The nutritional power is immense, and those health benefits are genuinely revolutionary. But I still believe its deepest, perhaps most enduring contribution is intellectual and frankly behavioral. By embodying and making visible these fundamental principles of ecology, chemistry through tracking pH, physics through measuring height, and the sheer necessity of patience over weeks, sourdough fermentation offers this unique, hands-on pathway to real scientific literacy. And it fosters a meaningful connection with the microbial world around us. It truly is an enduring educational model, turning basic flour and water into this incredibly powerful teaching tool for inquiry and critical thinking that can benefit generations of learners.
Well, both viewpoints certainly highlight the profound complexity and the really multifaceted benefits of this ancient yet remarkably simple mix of flour and water. There's clearly still a great deal more to explore within this whole fascinating frontier of fermentation.